on Saturday, and then all of a sudden someone turned the refrigerator on Sunday, and we had this awful, colder weather coming in again. But I think I'd predict it's not going to last too long. I think we'll get back to good spring weather again soon. Let's see, so you have the midterm number 4 coming up today in laboratory. And remember, it's going to cover, amongst other things, part of the genetics chapter, but not all of the genetics chapter. It's not going to cover sex-linked traits, which are the same as X chromosome, genes on the X chromosome. It's not going to have chromosomal abnormalities. And there was a part of the genetics chapter that we didn't cover called population genetics, and it's not going to cover that either. All those topics are going to be on midterm number 5, which will be in a couple of weeks. Which kind of brings me towards my next subject. What else are we going to be covering in here before the end of the semester? Well, here in the lecture hall, the next chapter I'm going to cover, as soon as I'm done with this one on genes, is called Viruses and Prokaryotes. And I wanted to point that out because I think in your syllabus it lists the next topic for the lecture as genomics. And so what I'm saying is I'm going to kind of reverse the order of these. This one's going to come first, and then we're going to do genomics. But that being said, I want you, so you should have this one printed up. We're probably going to begin it on Wednesday of this week. But that being said, start bringing with you to all lectures and all laboratories the handout that says DNA technology. Because I'm going to be working on this one sort of intermittently over the next few weeks. I might do a few paragraphs of it in one lecture, then do a few more paragraphs of it as a part of a pre-lab lecture, which we're going to sort of come back and forth to it several times, pretty much for the rest of the semester. As I mentioned in the genetics handout, the very last part of the genetics handout is called population genetics. And likewise, I'm going to do that segment. Well, I'm not exactly sure whenever I can find time to fit it in. So also keep carrying around your genetics handout. So be ready for me if I decide to say, okay, now we're going to do that population genetics thing that we never got to. All right, did I confuse everybody? You guys are too smart for me. Okay, well, just be sure you bring those with you all the time from here on in. All right, well, let's see. What we were essentially focusing on last week was transcription, which is when the messenger RNA is made from a gene. And the reason why we're giving so much attention to transcription is it's the major step where gene expression is regulated. Gene expression just means going from the gene to the actual protein in the organism. And when a cell is expressing a gene, it means making the protein from the gene. But sometimes a cell might not want to express a certain gene. And if the cell decides it doesn't want to express a certain gene, what it usually does is simply not transcribe that gene into a messenger RNA. So all the major regulation takes place in transcription, or at the transcription stage. And we looked at how prokaryotes do transcription, and then we moved on to how eukaryotes do transcription, and there were certain similarities and certain differences. Let's go over the way eukaryotes do it. If this represents, these two blue lines represent the double-stranded DNA in a eukaryote's, or a gene in a eukaryote, this will be the non-template strand up here, the one that the RNA polymerase does not ride along, and this is the template strand down here, the one that the RNA polymerase does ride along. This represents the first codon, the first 3 nucleotides that are going to represent the first amino acid. If you go upstream of the first codon, and when I say upstream, here's codon number 1, this would be codon number 2, codon number 3, codon number 4, codon number 5. If you go in the opposite direction, upstream of the first codon, there's a certain sequence of nucleotides of G's and A's and T's and C's up here called the promoter. And in prokaryotes, that's where the RNA polymerase enzyme attached to the DNA. The RNA polymerase is the enzyme that makes the messenger RNA. And in prokaryotes, there's a certain sequence up here in the promoter that the RNA polymerase had a binding site for, and that's how the RNA polymerase clicked on to the template strand to transcribe the template strand. In eukaryotes, it's slightly different. In eukaryotes, you find some sequences up here in the promoter, like there's one called the Tata box, named for this sequence you find right there, and there's another one called the CAT box, C-A-A-T, is a sequence you find in some eukaryotic promoters. But there's a lot more variability in eukaryotic promoters. Not all eukaryotic promoters have a Tata box, and not all have a CAT box. Whereas in prokaryotes, those promoters always had a certain sequence. They had to have that certain sequence because that's what the RNA polymerase bound onto. All right, so these boxes in the promoters of the eukaryotes, but more variability. And the enzyme that actually transcribes the mRNA is called RNA polymerase II in eukaryotes, whereas in prokaryotes it's just called RNA polymerase. And what's kind of interesting is in eukaryotes, the RNA polymerase enzyme, the RNA polymerase II enzyme, does not bind directly to the promoter sequence. And that's a big distinction between eukaryotes and prokaryotes. In other words, there is no binding site on the RNA polymerase II enzyme to latch onto the template strand. But in some way, it has to eventually become attached to the template strand so it can ride along it and make the messenger RNA. And here's how it works. There are some other proteins in the nucleus of eukaryotes called transcription factors, which are these things. And these transcription factors do have binding sites for the promoter sequences like the Tata box and the CAT box. So these guys do latch onto the promoter sequences. And once they've latched on, then they grab hold of RNA polymerase II and force it to click onto the template strand. In and of itself, the RNA polymerase II will not click onto the template strand.
Are there different transcription factors for different?
Yes, we're going to get into that more on this when we get into this chapter on genomics. But the short answer is yes. There's a whole bunch of different transcription factors that vary from one eukaryotic gene to the next, and that's what allows eukaryotes to have very complex expression of their genes, as opposed to prokaryotes, which it's a lot simpler. It's almost like the gene is on or off, and there's no middle ground. So I'm glad you mentioned that. The reason why we have this more complex system in eukaryotes is it allows for finer tuned control of gene expression than you find in prokaryotes. In any event, the transcription factors have latched onto the promoter sequences, and then once they're on there, they grab hold of the RNA polymerase II enzyme, and then they force it to click onto the template strand. And then once it has clicked onto the template strand, then you get transcription. It starts just making the messenger RNA, like you see there, and just riding along the template strand, making a complementary messenger RNA. And remember that any time a nucleic acid strand is made, it always starts off at the 5' end of that strand. That's always the beginning of an RNA or a DNA strand, is its 5' end. The 3' end is always the ending end, the finishing end. All right, so there's kind of the zoomed back view of the RNA polymerase II has been clicked onto the template strand. It rides along it, making the messenger RNA. Notice that the first codon is going to be right here, very near the 5' end of the of the transcript. But it's not, the first codon is not right at the 5' end. There's always a few nucleotides upstream before you get to the first codon in the messenger RNA. And those are called the 5' untranslated region, and we'll talk more about them later. Okay, so there goes the RNA polymerase enzyme. As it moves along the gene, eventually it's going to come to the last codon, which is always a stop codon, like you see there. And what happens, in fact, is the RNA polymerase II enzyme keeps transcribing right past the stop codon. It doesn't halt for the stop codon. Like that. Which sort of begs the question, well, what defines the end of the messenger RNA if the thing just goes right past the stop codon? Where does it end? Well, you might remember in prokaryotes, there was a sequence that was transcribed that destabilized the RNA polymerase, that made it fall off the template strand, and that's what brought the messenger RNA to an end. In eukaryotes, it works slightly differently. In eukaryotes, in the non-template strand of the gene, there's a sequence AATAAA, which notice is a little bit downstream of the last codon, of the stop codon. And that's called the polyA signal. When the template strand gets transcribed, that's going to end up now written in RNA language, AAU, AAA, but it's essentially the same sequence. And that poly-A signal, that AAU, AAA, is going to serve as a site where an enzyme in the nucleus clips the 3' end of the messenger RNA. In other words, there's an enzyme in the nucleus that looks for that poly-A signal in the RNA and snips it just downstream of there. Here's that enzyme. It's almost like a pair of scissors in the nucleus of the cell. Snip. And that's how the messenger RNA gets its 3' end. Just from being snipped downstream of the poly-A signal. Alrighty. Alright, now let's kind of zoom back a little bit here. Oh, there's that transcript coming off of there. All right, here's kind of a zoomed back view of the gene. There's the promoter where the RNA polymerase II got attached to the template strand, and then the RNA polymerase II ran along the template strand. The first codon is right about here, very near the 5' end of the RNA, but it kept transcribing. The last codon, the stop codon, is somewhere here near the 3' end of the mRNA. But then there were some more nucleotides. Then right about here was that poly-A signal. And remember, just downstream of the poly-A signal is where the RNA was clipped, like that. Okay, now the RNA transcript, just right after transcription, just like you see here, is actually called the pre-mRNA. It's not officially the RNA yet, mRNA yet, it's the pre-mRNA, or sometimes it's also called the primary transcript. And you notice a couple of facts about this. I mean, you probably know these already, but the pre-mRNA is as long as the transcribed region of the gene, because that's what it is. It was transcribed right off the gene. And maybe I should mention how the terminology for talking about how long a gene is. Genes come in different sizes because proteins come in different sizes, and genes encode proteins. When they're talking about how long a gene is, they often talk about how many base pairs it is. Because that's what genes are about, one base pairing with another base. Or sometimes they shorten that and just say how many bases long is the gene, how many nucleotide bases long is the gene. Now, genes can be several thousand, or typically several thousand base pairs long, and so oftentimes they talk about kilobases. KB. And base pairs are sometimes abbreviated BPs, but kilobases are 1,000 bases. So that's the terminology. The average gene is around 8,000 bases long, or put it the other way, around 8 kilobases in length. From the start codon here at the 3' end, the 5' end, to the poly-A signal there at the, or very near the 3' end, is generally on the order of 8 kilobases in length. Okay, so anyway, the pre-mRNA, which is also called the primary transcript, is about as long as the gene is from the gene.